Right now we're being recorded, so if you put something in the chat, if you put something in the Q&A, if you say something, you will be recorded. All of these recordings are being put up on Chestnut Chat. If you Google Chestnut Chat, our series is the first result that you're going to get. I'll also put a link down there in the screen. So I see John Harrington has a question in the chat about a New York Times article that came out a couple months ago. And John, we had an excellent Chestnut Chat that deals with that, where we had Bill Powell and members of his lab at SUNY ESF talk about all things related to that. So I highly recommend you check that one out. If we have time today, we might circle back on some of those more specific questions. But today we've got Bruce Levine, who is a grad student at the University of Maryland. And he's going to give a talk about plant pathology and give you sort of an introduction about what it means and what it is. Before we get started with Bruce, I want to turn things over to Lisa to give a welcome, and then we'll get started.
Hi, everyone. Happy Friday. We are delighted to have these Chestnut Chats, and we have several scheduled in the future. They've turned out to be a great engagement strategy for our community, keep people motivated and thinking about chestnuts during this pandemic. So I hope you all are well and safe. And I just wanted to say hi and take it away, Bruce. He's been a long time volunteer and researcher, has had a really storied career with the State Department, but has decided to devote some of his time now to chestnuts. So Bruce, take it away.
Sorry, Lisa, before we do that,
Yeah.
I just want to, really quick, so if you have a comment or something, maybe logistical with Zoom, please put that in the chat box. But if you have a specific question that you want to ask me or Bruce or Lisa, please use the Q&A panel that you can find that at the bottom of the screen. The Q&A allows us to keep questions and answers together. And just to remind folks and also introduce folks who have joined us since I made the announcement, in the Q&A section, you as attendees are all able to ask a question. You can answer a question should you feel that you want to do so, and you can also upvote questions. So the more a particular question has interest, then I'm more likely to ask that of Bruce. I typically hold all questions until the end, but if you're particularly confused about a particular thing and you want me to interrupt Bruce with to clarify something, I will or I can interrupt him. So just let us know. I'll be monitoring that. So will Lisa, and then Kendra Collins just joined us. She's our regional science coordinator up in Burlington. So we got a couple eyes on this to keep things coordinated. But with that, thanks for letting me interrupt you, Bruce, and take it away.
Thanks very much, and thanks for the introduction, Lisa and Sarah. I am going to share my screen, the slides for this. I think that's it. Okay, does that look like it worked?
It did.
Okay, good. All right, well, good morning, everyone. So today's topic, pathology for non-pathologists, is really going to be about what goes on in the relationship between microbes, and which are single cell organisms mostly, and plants at the cellular level, because that's where the action is. We're always talking about the genes for resistance and how we're trying to bring those over from Chinese chestnut to American chestnut, but you can't really understand what that means without understanding how plant immune systems work. And so that's really the focus of this conversation. And I'll start with a general overview geared towards non-scientists, and then talk a little bit about what this tells us about what's going on in chestnut. And I'll also say that everything I'm telling you applies for bacterial diseases, fungal diseases, oomycetes, even nematodes and some insect type diseases, but not necessarily for viruses, which is a bit of a different story. Anyway, let me see, how do you do this? Okay, so to begin with, we have to talk about what we mean by immunity. And when immunity has been in the news a bit lately, when you talk about human immune systems, we're always thinking of how our body can make antibodies against pathogens that our bodies recognize as something that's not supposed to be there. And it's very effective, and You know, we use vaccines to trick our bodies into developing immunity to things we haven't been exposed to. That's what we think of when we think of immune systems as human beings. But actually, it's this great system, and it sits on top of something that's a different system called innate immunity. So the ability to deal with a pathogen your body has never seen before is adaptive immunity. But the innate immunity is something that's built into every cell. We have in the human genome a couple hundred genes that encode for various proteins whose function is to defend the cell and make it an inhospitable environment for microbes. And it's super effective. We don't talk about it very much because it usually just works, and it's broad spectrum. But if you didn't have it, there'd really be no difference between a living animal and a dead animal, or a living plant and a dead plant, in terms of how quickly they decay just under the pressure of the bacteria and fungi that land on them every second of every day. The innate immune system is really important. And then our adaptive immune system on top of that helps us deal with those pathogens that have learned to overcome our innate immunities. Okay, but for plants, I want you to get the adaptive immunity out of your head, because they don't have it. They actually do in the viral area a little bit, but we won't talk about that. What they have instead is a very complex innate immune system, okay? And just to illustrate how effective that is, I have this picture here taken in our lab of two Arabidopsis plants that have been inoculated with a kind of powdery mildew fungus that isn't supposed to grow on powdery mildew. It's not adapted to grow on powdery mildew. The plant in the upper left is the wild type plant, and the one that's infected in the lower right has the same exact genetic background, but has 3 genes deleted. And these were genes that were suspected as being part of the defensive system. And you can see how 3 genes out of tens of thousands of genes can make all the difference. I'm going to use a fortress analogy to explain how this innate immune system in plants works. And there are 3 tiers to it. The first one is called constitutive defenses, and the next 2 are induced defenses. And when I say that there's tiers, it's not like first one happens, then the other, then the other. They are all occurring simultaneously, but this is a way that we categorize them as we model how plants are resistant to disease. So I want to imagine a plant cell as a fortress. You've got a building in the middle here where the king and queen live, you know, the nucleus and all the juicy stuff in the cell, but it's protected by this big wall around it. It can communicate with the outside world through windows and doors, but the windows are high and inaccessible. The door has a drawbridge and a portcullis. There's a moat around it to keep bad guys from getting close to the fortress. There's alligators in the moat, so if bad guys do get in the fortress, they get attacked by the alligators. There's spikes, you know, maybe to keep even further away from the castle, right? And these features here do a really good job keeping unwanted personnel from entering the fortress. And they're there all the time, whether or not bad guys are around, okay? So that's why we call them constitutive. They're part of the cell's very makeup, and they're always on, basically, okay? And they're encoded by genes in the plant cell, as we'll see. So the next 2 tiers are the induced defenses. And induced defenses are for when, despite your constitutive defenses, a pathogen manages to get access to the cell. And so the cell has to respond with an escalated defensive response. And this has 3 components. It has to recognize that a pathogen is there. It has to, whatever recognizes it, has to signal the defensive response. And then there's the defensive response that actually fights the pathogen. So I represent these here with a sentinel who's on the lookout for bad guys. And if he sees them, he blows his trumpet. When he blows his trumpet, the archers come out to shoot arrows and the soldiers get ready to fight hand to hand. These are 2 different defense responses both triggered by the same signal. And that's the same kind of thing that happens in a cell. Now you can imagine if you're in this castle and suddenly the trumpet blows and all these archers and soldiers are running around, the castle's going to have to stop business as usual. There's going to be no more making tapestries or having feasts or witch burnings or whatever they normally do. That all has to stop so that the defensive response can happen. And a similar type of reallocation of resources happens in the cells. So let's take a look at what goes on in the cells. It's a very simple model of a plant cell, okay? Here's your cell membrane going around the outside in green, and all the juicy stuff that the cell has to protect is inside that cell membrane. Here's your nucleus. Around the whole thing is a secreted cell wall, which is a porous and very strong barrier, kind of like a wicker basket. But it's not a dumb barrier. It is dynamic. It gets softened and stretched and remodeled and constantly repaired as the cell grows or gets damaged or whatever. But the cell wall is the central element of constitutive defenses. Not only does it provide a physical barrier that is constantly reinforced and repaired by secreted enzymes, but it also contains lots of antimicrobial chemicals or chemicals that repel water, things like that. And on top of the cell wall, other constitutive defenses would include other compounds that the cell secretes to the outside world. And these things can include antimicrobial chemicals of various types. It can also include simple things like waxes that may accumulate on the surface of the bark or the leaf and cause water to run off so it doesn't remain in contact with the cell membrane and with all the microbes that it may carry with it. Those are your constitutive defenses, and they're very effective. So when we think about what kinds of genes, the genes behind the constitutive defenses, these are the genes involved in the production, delivery of the cell wall and its components, the protective secretions like waxes and things like that, and then the production of antimicrobial compounds. And I highlighted that one in red because that's where, in my very humble opinion, because really there are many people who are better versed in this than I am, that's where I think a lot of the resistance from Chinese chestnut comes from. Now, this little cartoon here is just to illustrate the definition of a pathogen. It is a well-known scientific fact that microbes are orange ovals with smirks on their faces, and they want the nutrients inside living cells so that they can grow and reproduce. But they're prevented from reaching those nutrients by the constitutive defenses, which are a metaphorical wall, right? But one of these microbes out here actually has an adaptation up his sleeve. So he's smiling. He's got this ladder. He can get over the wall and get access to the nutrients. That's what makes him a pathogen, okay? Now, in the real world, what this is these types of adaptations can include specialized enzymes that degrade the protective layers of wax or the cell wall components itself, or toxins that weaken or change the cell's behavior or even kill it. It can also include specialized growth structures that physically push their way into plant tissue. And we can illustrate this in action with this picture. So these are some chestnut blight fungus inoculations I did on chestnut stems a couple of years ago. These stems, although I cleaned them with bleach, they were covered with various types of saprophytic fungi, which means fungi that eat dead tissue, on the surface, all different kinds of fruiting bodies and things. They got really moldy in the refrigerator. And then I inoculated them with the chestnut blight fungus, and a few weeks later peeled off the bark to see the extent of the canker that formed. All this brown stuff here on the left is necrotic chestnut tissue where the chestnut blight fungus has killed the cells and is digesting them as it spreads outwards, okay? And it's doing very nicely. On the right is a chestnut stem that had died before I inoculated it, I guess, or shortly after. And the cambium layer was all dried out. The cells were no longer alive. And all that fungus that was on the surface started growing underneath. And I can't tell where the chestnut blight begins or ends and the other fungi begin and end. It's a free-for-all in there. And what you can see here is why it's good to be a pathogen, because a pathogen, like on the left, can use the defenses of the living plant cells to keep the competition out. And because it has special adaptations that allow it to survive despite those defenses, it's got the place to itself and can feed and reproduce as it wishes. All right, so just as in the case with the fortress, you will get the occasional bad guy who gets through your constitutive defenses, and you need a plan B. In the fortress, that was the sentinels and the archers and the soldiers. In a plant cell, this is kind of what it looks like, again, oversimplified. The sentinels are these squiggly things that are floating, literally floating in the cell membrane, and they're called pattern recognition receptors, or PRRs. And they're proteins encoded by genes, and they have a part that sticks out to the outside world and a part that sticks in. And they come here, I have them in 4 flavors. In reality, you would have dozens or hundreds of different types of PRRs on any given plant cell membrane. And they have affinities, molecular affinity for particular molecular patterns that are associated either with pathogens or with damage caused by pathogens, okay? So these are your sentinels. So in this case, the blue one is sensitive to something that fungi typically have. For example, chitin, which is what fungal cell walls are made out of. The bacterial one may be sensitive to a protein called flagellin, which ends up sloughing off of bacteria when they wiggle their flagellae. Oomycetes secrete other things that trigger other PRRs. And damage, like if a fungus is dissolving the cell wall, the byproducts of that dissolution can trigger the damage-sensitive PRR, okay? And when the outer part of the pattern recognition receptor bonds to the thing for which it has an affinity, the whole protein undergoes a conformational change. It means a change in shape that basically shakes loose an ion on the inside of the cell, a phosphate ion. And that acts as a signal. It triggers a cascade of phosphate ion signals that go all the way down into the nucleus, where there is a receptor or receptors that receive that. And then that triggers changes in the way genes are expressed in the nucleus, which is your defensive response. So the nucleus turns off some of the routine housekeeping genes in order to save energy, and then launches a defensive response. That can include an enhancement of activities that you would see in constitutive defenses, like cell wall reinforcement, et cetera. It can include a signal for the cell to divide more often so that the tissue in which the cell resides starts to build up and thicken to put a barrier between the pathogen and the live tissues of the plant. It can include something called an oxidative burst, which is a release of reactive oxygenated chemicals like hydrogen peroxide that are very toxic to microbes, or the release of other antimicrobial compounds, either the same ones that are produced in constitutive immunity but at higher levels, or other ones that are not normally produced. And it can also involve a signaling to neighboring cells so that those cells can prime their immunity without having to have contact with the pathogen first. And this whole system that I just described is called Pattern-Triggered Immunity, or PTI. And it responds to broad categories of microbes, okay? Either bacteria, fungi, oomycetes, et cetera. So what types of genes are in Chinese chestnut that we're trying to bring over? Well, first of all, they may have slightly different pattern recognition receptors who are better optimized to recognize chestnut blight fungus. I don't know that to be the case, but it's possible. It's likely that the genes that regulate the signaling and the gene activation in the nucleus are pretty much the same, but they could be different. They could be more responsive. But I think that the real difference is in the response itself, which we've all observed seems to be more robust, quicker, and more intense in Chinese chestnut than it is in American. And you can see it with a greater thickening. And as I'll point out later, there's evidence of more antimicrobial activity. Okay, so PTI works really well in protecting plant cells. And so any pathogen worth its salt is going to have to find a way to shut that down. It's going to have a hard time getting in the cell as long as PTI is working. And so the way pathogens deal with this is through the use of effectors, which are chemicals that suppress the defensive response of the host. There's no other chemical definition for effectors. They're not all similar to each other. They're not, they don't all have common features. But what they do is they have a common function, which is to screw up the defensive response, and particularly the PTI of the plant. And they can act on the outside of the cell, for example, by camouflaging the pathogen or blocking the receptor from noticing it. They can act inside the cell and screw up the signaling between the pattern recognition receptor and the nucleus. They can act in the nucleus itself and prevent the recognition of the signal or prevent the defensive response. And they can even act on the way back out on the response itself, sometimes hijacking it in order to turn it against the cell itself and kill the cell. The various ways they work then are weakening the plant through toxicity, blocking recognition, disrupting signaling, etc. And effectors tend to be very host specific. And what I mean by that is while all plants have cell walls and they're pretty much the same, and all plants have pattern recognition receptors, those receptors aren't likely to be the same from one species to another. And in order to suppress their activity, you're going to have to match them perfectly so that your effector interferes with that molecule. So it's going to have to be very specific to the actual sequence of amino acids in that protein, which means the gene that encodes the effector has to be very specific to the gene that makes the pattern recognition receptor or whatever it is that the effector is supposed to interfere with. And so effectors tend to be very host specific. Effectors that work on chestnut very effectively will not work on a maple tree, as in this picture, where I inoculated a maple with the chestnut blight fungus. 6 months later, I came back and all I could see was a small scar from where I punched the hole. But there was no evidence of infection whatsoever. I couldn't re-isolate the fungus from this spot. There are lots of other fungi, as you can see, growing on this maple tree, but they're not chestnut blight. And if I put them in a chestnut tree, I'm not sure they would grow. They may be specific to maple. So the effectors are very host specific. They're produced by particular pathogens and for use in particular hosts. And plants also have a plan for dealing with them, which makes up our third tier of immunity, effector-triggered immunity. We have pattern-triggered immunity, responds to molecular patterns associated with broad categories of microbes. Effector-triggered immunity responds to effectors secreted by specific types of microbes, and it's much more species specific. And there are really two types. The genes that of immunity, the genes that encode for effector-triggered immunity are called R genes in the business, and they do generally fall into two categories. One is that they're decoys, okay? Like these, I kind of drew them as semicircles. And what they do is they bind to the effector. So in a sense, the effector is reacting to it the way it would to its actual target. And then once it's bound to the decoy, it's neutralized. It can't bind to the target anymore. And so that's a way of getting the effectors out of the picture. Others that I represent here as blue squiggles are receptors similar to the pattern recognition receptors, except they're free floating. Some work outside the cell, most work inside the cell. And just like with PTI, except they sense the effector and they send a signal to the nucleus that the cell is under attack, with the exception that they're not, they're binding to the effector, not to some broad-based molecule from the pathogen. And their signaling pattern is similar to that of PTI, but it's separate. In experiments where pathologists have disabled PTI in certain plants, they find that the ETI signaling still works. So it's like a backup alarm system. And it triggers a defensive response. The defensive response is essentially similar to PTI, as far as we know, but it's it's at a higher level or a higher volume. And it can also trigger something called the hypersensitive response, which is when the cell basically commits suicide, which is a very effective defense if your pathogen needs to interface with a live cell in order to extract nutrients, as in the case of powdery mildew. But it's not a good defense with chestnut blight because the chestnut blight fungus wants the cell to die. It cannot eat a live cell. It's a different type of pathogen. And it's even possible, I don't have any evidence for this, except that this occurs in other systems. It's possible that the chestnut blight fungus actually has effectors that trigger the hypersensitive response in the plant cells in order to facilitate their rapid death. Again, that's just a hypothesis, but that does happen in other systems. So that's your effector-triggered immunity. And the types of genes that would be associated with that would be the genes that encode the proteins that make up your receptors and decoys. And those are what we would want to bring over from Chinese chestnut into American. In terms of the signaling, the gene activation pathways, and the response itself, they're probably not too different between the two species. So, sorry about that transition. That leads us to the question that now that we have a basic picture of what the plant immune system looks like, what do we specifically know about the genes for resistance in Chinese chestnut? And the answer is we don't know a lot. We have not identified a single, and we know there are several, responsible for that resistance. There are a couple suspects, but it's not as easy as it sounds to actually find these genes, especially in slow-growing plants like trees. But we do know something. One thing that we know is that the constitutive defenses in the bark of Chinese chestnut seem to be superior to that, to those found in American chestnut. There were a number of papers done in the 70s and 80s where people actually isolated specific proteins or other compounds and showed that were found uniquely in Chinese chestnut and showed that they were very inhibitory to the fungal growth for that particular species of fungus. In this picture, you see two petri dishes made with a brown medium, which is a mixture of tea made from chestnut bark and agar, okay? And then we inoculated it with the same strain of fungus on the same day. The one on the right is the Chinese, the one on the left is the American. And as you can see, the Chinese is very restricted. It hasn't even reached the edge of the dish after 11 days, whereas the American's already, the fungus is already crawling up the sides of the dish. If you could get this fungus off here and weigh it, you would find it probably weighs less with the Chinese. This excess fuzziness you see is called aerial hyphae, which is what fungus does when it doesn't like the substrate that it's growing on. It puts these hyphae into the air in order to find another substrate. And there's less of that in the Americans. The most significant thing I see, though, is the American is doing this very normal petri dish growth of a daily cycle, light and dark. When, you know, as the cycle changes, it puts down a layer of differentiated cells that grow into fruiting bodies. So this one is already starting to produce spores. And we see none of that in the Chinese. So the development of the fungus is being inhibited by something in Chinese chestnut bark, or maybe several things. So that's the constitutive defenses. Here's a picture demonstrating PTI. And this is a Chinese on the left and an American on the right that have both been inoculated with the chestnut blight fungus. And the Chinese heals up beautifully. And you can tell something's going on. It's definitely swollen in there. So there is a defensive response that's happened. The tree is healthy. There's no fruiting bodies. If you could cut it and look at it, sideways, you would see a thickened layer, but you wouldn't see the fungus down to the living layer of cambium tissue. And that's why the tree is relatively unaffected. On the American, however, the fungus has grown without impediment. Not that the tree hasn't tried. Where these arrows are, you can see little ridges that represent the attempt to build a barrier, but the fungus was able to just blow right through those. I believe because the fungus produces effectors that shut down the PTI response or weaken it, and the American just can't do what it can normally do with other fungi, which looks like this. So this is an inoculation of Pestiloteuthis, a mild chestnut pathogen for which American chestnut has a high tolerance. And 4 months later, you see a really nice, big, thick canker, just like you would see from chestnut blight fungus on a Chinese chestnut. And there's a little bit of fungal mycelia still, but it's mostly gone. So there's nothing wrong with American chestnut's ability to do PTI. It's not inferior to Chinese. If it were, the tree wouldn't have been so successful until the blight came. The problem, I truly believe, is in the effector-triggered immunity. It just doesn't have effectors that respond to, it doesn't have R genes that respond to the effectors produced by the chestnut blight fungus. I'm going to end on this last slide with the small stem inoculations, just because they show the difference more clearly. Again, both trees were inoculated on the same day, Chinese on the left, American on the right. And you see they're both badly affected by the fungus, but the Chinese is recovering. It's thickening here. It's stopped the canker from making it all the way around the stem. Even if you look at the margins of the infection, you can see the fungus and the tree are pretty evenly matched. They're fighting cell to cell. And this Chinese will eventually slough off this infection. The American, you can see again, there are little ridges where the tree attempted to stop the infection, but the attempt just wasn't strong enough, probably because the effectors from the fungus suppressed it, and this tree wasn't able to detect the effectors and enhance its defensive response. The tree's actually dead from this point on. So I'm going to stop there and open up for questions, if that's all right.
Yeah, that's great. Thanks, Bruce. We have one question so far, and hopefully while we're looking at that question, we'll have other folks ask some more questions. This is from Louise Alcott, and I'm guessing Mike too. Hi, Mike and Louise. How much of a role do you think plants' fungal associates, for example, mycorrhizae, et cetera, play in bolstering immunity to pests?
Okay. Well, that's actually, that's a huge subject of research. It varies a lot from species to species, and the particular mycorrhizae. So for those who don't know, mycorrhizae are fungi that interact with the plant roots in a symbiotic way. And they grow on the plant roots. The plants give them a lot of carbohydrates to eat. And in exchange, what the fungi do, among other things, is they basically extend the surface area of the root system. And they're especially useful in getting phosphate out of the soil for the plants. But they also seem to suppress the growth of other fungi. It's possible that some mycorrhizae, because they're also made of fungi, do trigger an early PTI in the whole plant, because I said there was signaling from cell to cell. And so the plants are better primed to fight fungi. But I think the main benefit is it keeps other root pathogens away, physically blocking them, and also by improving the nutrition of the plant.
Cool. I have a question. There's a couple more on here, but I have a question for you for myself.
Yeah.
When I first started, there was a lot of talk about gene for gene interactions, and you mentioned R genes and things like that. Would you be able to speak to that sort of at a broader level, and maybe how that would affect chestnut and chestnut blight specifically?
Sure. So the gene for gene interaction idea was, they noticed that there were certain genes in certain pathogens that if you deleted them, like the gene that makes oxalic acid in chestnut blight fungus, if you deleted them, the pathogen becomes much less virulent or avirulent. And so they confusingly call these avirulence genes, even though they're really virulence genes. And then they discovered these things were actually effectors, genes that encode for effectors. Now, I don't know if it depends on your definition whether you would call oxalic acid an effector, because it's not a protein, but very often they are proteins. And so the pathogen produces some effector, and then they discovered that some plants have genes that interact with the effector and prevent it from doing its job. Okay, those are the R genes. So people started looking for those and finding them in many different pathosystems. And what they noticed was there's a lot more effectors out there, almost an unlimited number, and we keep finding more each day, than there are R genes. And so they said, that's weird. How is this working? And one of the explanations is something called the guard hypothesis, where the R genes aren't actually interacting with the effector. They're actually guarding the thing that the effector attacks. And so if anything happens to that signaling molecule or whatever that molecule is, the R gene then triggers. And so it can protect against many, many different effectors as long as they attack that, that site. But in terms of chestnut, we're not in a position yet we haven't identified a single effector from the chestnut blight fungus, unless you want to expand the definition of effector to include things like oxalic acid.
Great, Bruce. So before I ask some more questions which are related to that topic, Bruce, you're doing some good segues here. Steve Jeffers, I think you know Steve at Clemson, says, great job explaining the different host plant defense mechanisms. You made a complicated system understandable. And I agree, it's incredibly complicated. So thanks for giving that analogy with the castle walls and things. I think is a perfect way to describe it. You brought up oxalic acid, and so I have a couple people here who are asking questions about that specifically. So Dean Tully asks, how does the oxalic oxidase gene confer immunity?
Okay, so oxalic acid, this goes back to the work of Bill Powell and others, going back many years. They found that oxalic acid is necessary but not sufficient for the chestnut blight fungus to kill American chestnuts. And they did this by deleting the oxalic acid encoding gene, which, thank God, there's only one in the chestnut blight fungal genome. And then they took the fungus with that gene knocked out, you put it in a petri dish, it grows perfectly normally, you put it in a tree, and it doesn't grow well. It's not pathogenic. Okay. And so they said, aha, this is a key thing. It is a key thing, but just having oxalic acid doesn't get you access to a chestnut tree. A lot of fungi, Dutch elm disease, for example, produce oxalic acid. There's a fungus which is practically the poster child for oxalic acid production called Sclerotinia sclerotiarum. It grows on all kinds of herbaceous crops. And I took some of that and inoculated it into Chinese and American chestnut trees 2 summers ago. And it did nothing at all. The trees healed right up. And I couldn't isolate it. Even 2 weeks later, I couldn't re-isolate that fungus from the tree. It just wouldn't grow. So without oxalic acid, chestnut blight cannot grow well in an American chestnut, but another fungus with oxalic acid isn't going to grow in chestnut unless it also has other effectors. What the role of oxalic acid is, there are a number of different theories, and they may all be true. One thing is it's toxic. Another is that it lowers the pH of the interface between the pathogen and the tree, so that the enzymes, like the enzymes that break down cellulose, they work better in an acidic environment. So it tips the scales in favor of the fungus on the battlefield, if I can mix metaphors. And then I read something recently, I can't remember whether it was tobacco or cotton, another crop, they used CRISPR to take out the oxalic acid gene from some other fungus. And as a result of that, they were able to figure out what it was doing was it was delaying the oxidative burst. When the fungus, comes in contact with the plant cell and the plant triggers PTI, that oxidative burst I talked about is really important. And somehow oxalic acid was preventing that from happening. And so that conferred defense. Now, the question was, how does the OXO counteract it? OXO is an enzyme, or the gene from wheat produces this enzyme, OXO, and OXO breaks up oxalic acid. It turns it into something else, I think hydrogen peroxide, carbon dioxide or something. And so it just neutralizes the acid before it can do harm. And the transgenic trees just constitutively produce it. How do I know that? Because the promoter that they used for the gene is a constitutive promoter. And interestingly, if Bill Powell's there and I am wrong about this, please chime in, but I think they found, at least in Chinese chestnut, that there is a similar OXO encoding gene already in the Chinese chestnut genome, but I don't think there's any evidence that it actually comes into play when there's a chestnut blight infection yet.
And I don't see Bill on here. I see Linda on here. Maybe she'd be able to pipe in on that. But I do want to caution against the term immunity, which Dean used in his question. I don't think OXO confers immunity. We use the term blight tolerance. The transgenic American chestnut is not completely immune to chestnut blight, even with the OXO gene. It can still get the blight. And so that kind of leads into Dan Shadler's question, which is, how do transgenic American chestnuts respond to inoculations similar to the one you showed? So Bruce, you're showing this picture right here of the SSAs. Can you speak to how the SSAs, how the transgenic trees or the OXO trees respond to the SSA inoculation?
If Linda's on there, maybe she can do that, because I've actually never done it. I've seen pictures.
Yeah, I don't know if I can give her the floor. Maybe she can. Let's see. Linda, I'm going to put you on the spot if I can find you here. Linda, are you able to talk?
No?
Can you hear me?
Yeah.
Yes, we can hear you. Hey, Linda, we have two questions. So one, yes, we can hear you. Can you hear us? Yes, I can hear you, Linda. Maybe you can't hear us. Linda?
Okay, can you hear me now?
Yes, can you hear us?
I can now, yeah.
All right, excellent. Hey, Linda, we have two questions for you. So one, can you speak to Chinese chestnut having an OXO-like gene? And two, how do the transgenic American chestnuts respond to the SSAs?
Okay, so I'm not in the molecular aspect of the transgenics. So I don't know about the molecular parts. I believe the Chinese chestnut, I want to say it's a German gene or something where, yeah, there's something similar in Chinese to the OXO gene, but again, I apologize, I'm not the molecular scientist. So.
Yeah, German is OXO.
Right, and I think they found that in Chinese chestnut, but I'm not 100% sure about that.
And the other question was, what does it look like when you inoculate a small stem of a transgenic tree with the OXO gene compared to these two pictures on the screen?
So it kind of has the same response as the Chinese chestnut. It does get the fungus in it, and it does start fighting it off. Mm-hmm. You know, we haven't had a lot of stems to inoculate, but the ones that I've seen seem to have a very similar response to the Chinese chestnut.
So yeah, so I just want to make that comparison or make that distinction between immunity, which would be where the fungus is unable to infect, or if it does, it pushes it out almost immediately, versus tolerance or resistance. And tolerance is a type of blight resistance, and I know these definitions get really squishy, but tolerance means that it will get infected, but then the tree will be able to fight it off using the various defense mechanisms that Bruce has covered. Thank you, Linda. I appreciate you letting me put you on the spot like that.
No problem.
Yeah. This is really interesting. I love this topic of cruddy bark. And so Paul asked this question. I'm going to introduce the term cruddy bark. This is usually used in consultation or in conjunction with what we think of as hypervirulence or biocontrol. And hypervirulence is where the virus infects the fungus. But there could be other mechanisms that are at play too with the cruddy bark. So Bruce, can you speak to that?
Sure. So a lot of people have looked into this so-called cruddy bark question. And what that refers to is cankers that get really ugly really fast. And we find that we can isolate from them lots of different types of fungi, even bacteria. But the tree seems to suffer less with those cruddy, ugly, rusty looking cankers. They're like black cornflakes glued to the tree, you know. But nobody really knows what's going on. For example, if hypovirulence, which is a viral infection of the fungus, sets in, you very often get a cruddy bark type, that's beautiful, kind of reaction. This tree, this picture, by the way, you're showing is Tom Scrivener's tree in Hagerstown, Maryland. And this tree, it looks like it's wearing a sweater made out of cruddy bark, and the arms that stick out of the sleeves are the branches where suddenly the cruddy bark stops and normal bark starts. And each year it creeps up a little more. But the tree's like 25 years old. But one of the things about this tree is it has completely screwed up random branchiness. It is the worst form of any tree I've ever seen of any kind, including like a Bradford pear. And I talked to my advisor about this tree once, and he said he thought it may have a mutation in its one of the enzymes. What's the big one? IAA. There's a plant hormone that regulates branching and apical growth, but that hormone is also involved in the deposition of vascular tissue in the plant. And if there's something wrong with that hormone, you could end up with screwed up apical growth, as we see in this tree, but also accelerated vascular tissue production, so the tree is able to repair itself faster when it's under attack by blight. And so this tree may survive blight, but it would never survive in the forest because the shape is so bad, even if that were true. And so this may not be a desirable mutation to pass along. In other cases, cruddy bark may be the infection of something like trichoderma or another fungus that does suppress the chestnut blight fungus, but we haven't really identified any of those for sure yet. So I think the answer with cruddy bark is we just don't know.
Bruce, is this the Corrigan clone, or is this a different tree?
This is an old ACCF tree that Tom Scrivener got like 25 years ago.
And I'm asking because the Corrigan, I know the Corrigan clone, which is an ACCF tree too, when we did some back crosses with it, they then expressed a lot of this similar phenotype. So that it's one of the first times that I saw this be under genetic control rather than being other fungi or bacteria or, you know, whatever fighting with the fungus. So I think this is a particularly interesting case. Yeah, okay, so that takes care of cruddy bark. Let's see, another question. Okay, so what is known about the American chestnut response to Phytophthora, which is apparently mostly lethal to them?
Yeah, so what's known is that American chestnut has no observable resistance to Phytophthora cinnamomi, whereas Chinese chestnut has a fairly high degree of resistance to it. Unlike chestnut blight, though, that resistance seems to be controlled by fewer genes, or at least there's a few very important genes. And so we have managed to capture resistance to Phytophthora in some of our hybrids from the Chinese. And it's much more, it's not exactly an all or nothing thing, but it's much more like that than the blight resistance seems to be.
And I want to thank Steve again for coming up with a better definition or a better job than I did of immunity. So Steve says, immunity literally means no disease. Resistance and tolerance both imply some level of disease. There's a continuum between highly resistant and highly susceptible, and the degree of resistance depends on the genetics of the host plant. And I think that's really, really well put. So thanks, Steve. Let's see. So Tom Green asks, you mentioned plants defend against some insects in a similar way. Can you say more about the similarities and differences between plant response to microbes and insects, and give any examples of where insects where the response is similar?
Sure. And it's not even just insects, but even animals like acacia trees in Africa. I mean, if a bunch of gazelles come up to the acacia tree and start nibbling on it, those damage receptors, remember I showed the PRRs can include damage receptors? Damage receptors will trigger a defensive response that causes the tree to start producing more tannin in its leaves. And at the same time that happens, the trees also give off ethylene gas, which is a very common plant hormone. And that gas gets carried on the wind, and the surrounding acacia trees pick it up, and they start making more tannin. And this was discovered a few years ago in South Africa when a bunch of gazelles or something started dropping dead for no reason. And it was because they had overstressed a couple of acacia trees, and then all the acacia trees became toxic. Okay, so that's PTI happening, plus the sort of signaling between trees. It's very interesting. When you get down to things like nematodes, they are almost like microbes. They actually secrete effectors, and they can cause the plant to make changes. Someone's calling me, believe it or not. It really works just like it does with microbes. I'm gonna put this phone in my drawer. Oh, it's not the phone, it's the bass that rings. Damn. Okay, just ignore it.
Okay. So I've got two more questions here. I have any chestnut blight pattern, three, okay. Have any chestnut blight pattern recognition genes been identified, and have they been transferred to other organisms?
Okay, so remember that the pattern recognition receptors are part of the plant, not the fungus, just from the way the question was worded. And no, not specifically, although, I mean, well, yes and no. So when you sequence a genome of a plant, like American chestnut or Chinese chestnut, so you get all this sequence, right? And then they stitch it together and they come up with this long sequence. And then they go and look for the genes. And first that's done with software, and the software goes through and they have all these algorithms that kind of predict genes, and then you run some Python script to compare those genes to huge databases of genes from other species. And it'll tell you, oh yes, this is exactly like, or very similar to a pattern recognition receptor that was found in roses by, some Japanese scientist or whatever. And you start to populate your genome map with these annotations. Whether you're 100% sure that those are right or not, well, you're not. Certainly you could do that with either Chinese chestnut or American chestnut, and it would be easy to identify pattern recognition receptors because the proteins have a very specific architecture, and you'd be very unlikely to be wrong. You might be wrong about whether that gene is actually expressed or not, but you would be able to identify it. But to my knowledge, no one has ever really done it. However, in any genome, you look at these genomes online at the websites where they're hosted, and you click on the gene, and then always a box will come up that will give you annotations that tell you, it's called gene ontology, what the gene is likely to do based on its similarity or lack thereof to other known genes in other species.
All right, a somewhat related question. Is there a difference in the mRNA response between Chinese and American chestnut? Can we introduce resistant genes that are present in Chinese response and introduce them with vectors to the American chestnut?
Is this question from Moderna? Is this Anthony Fauci?
An anonymous attendee.
Oh, okay. Yeah, it is Anthony Fauci. Okay. No, I don't know the answer to that. I would assume not. I mean, this is both pretty routine gene expression, and when you're, but there have been attempts in plant pathology to try to influence or induce resistance to disease using RNA, but mostly that is done through something called host-induced gene silencing. And it uses remember I said there's an adaptive immune function in plant cells that works for viruses, but I wasn't going to talk about it because it's not relevant to this topic, and this is it. So plants do have a way of recognizing double-stranded RNA, and since that's not normally found in plants except very transiently, the plant, knows that there's a virus present, or it thinks that there's a virus present, and it has a whole elaborate defensive response to that can result in seeking and destroying any other double-stranded RNAs that have the same pattern. So if a virus comes in, they sense the double-stranded RNA and go and seek and destroy those exact sequences of RNA and prevent the virus from being expressed. And so you can trick plants into doing this against anything. So you make a double-stranded RNA, for example, against some critical gene in chestnut blight fungus, let's say the oxalic acid gene, and you prime your plant with that. So as soon as it sees that gene being expressed, and by the way, the double-stranded RNAs, they move freely between host and pathogen, which is really convenient. And they have like a free pass. They just go right across. And so it gets into the fungus and will start to silence the expression of that gene in the fungus. Host-induced gene silencing is what this is called. It's a fiddly approach. If there's a better approach, like a naturally occurring resistance gene, you want to go with that. But they've used it, for example, they've made host-induced gene silencing banana trees that don't get Panama disease, which is like a big deal. They've made host-induced gene silencing wheat that doesn't get fusarium. If you've eaten a papaya grown in the state of Hawaii, that is a host-induced gene silencing tree that is defended artificially against papaya ring spot virus. But I don't know anyone who's worked on host-induced gene silencing for chestnut, because chestnuts are very difficult to transform. And
then just to answer that question, like is there from a higher level of, or is there an attempt to try and get Chinese chestnut genes into an American? There is. There's attempts to try and figure out what are the genes in Chinese chestnut that actually confer resistance, and that's a big unknown. We don't know exactly what those genes do, exactly where to find them, and to put them in an American chestnut. Is it possible? Yes, conceivably, but there's a lot of work that needs to be done to get there, and that work's being done. But it's a long road versus, say, OXO, which is already created.
Let me just, can I add to that?
Absolutely.
Yeah, so first of all, this idea of, finding the gene that you want to put in there is actually a big deal. You always hear about it, but it takes a long time, even in plants like Arabidopsis, where you can get 4 generations a year and you can self-pollinate them. So you're not crossing and diluting your genomes every time. It can take years and years and years to actually locate a specific gene. And that's why it's taken us so long with this. If you could move those genes, if we develop a way to use CRISPR on chestnuts, we could actually edit, say, the American chestnut version of gene X to look more like the Chinese gene. But where I think in the foreseeable future, where the benefit of identifying specific Chinese genes of interest will be in screening our trees. So let's say we discover that gene ABC, the Chinese version, results in much more resistance than the American version of that gene. You can easily design a genetic test, and you take a little sample of a leaf, you grind it up, extract DNA, run the test on it, and you can find out, have I inherited the Chinese version or the American version or one of each? And you can do that with all the seedlings that you grow. And then you can say, I'm only going to breed with the ones that have 2 copies of the Chinese, or I'm only going to breed, you can use it for selection. And I think in the foreseeable future, that's the first place that this is going to be useful. Those tests are fairly easy.
And then just sort of along that, because it came out just this morning, hot off the presses, the reference genome for Chinese chestnut for the Venuxum cultivar that we're using in TACF, that was published this morning. In Tree Genetics and Genomes. So I put the link there if you guys are into that. That is about a 20-year project in the making, and almost 30 authors on that paper. So if you're into reference genomes, that is a big one. So I hope you can check that out. So we have one last question. I know we're a little over time, but I want to make sure we get to this one. Richard Bray asks, how does the chestnut blight actually penetrate the bark of a healthy, mature wild tree? And what is the risk of transferring blight in the search of wild trees?
That's a good question. So unlike powdery mildew, chestnut blight does not have these high pressure structures that can push their way into plant cells. The fungus will grow on the surface of the bark, and it will spread out, and it'll live off of the dead tissue on the surface until it can find a crack, a naturally occurring crack. And then the hyphae grow into that crack. And they grow under enough pressure. I mean, they're tiny and you wouldn't feel it, but if you convert it to pounds per square inch, it's like 1,000 pounds per square inch of pressure. And so the hyphae of the fungus will grow in between the plant cells. If it's a harmless fungus, they'll share the space. If it's harmful, they'll kill the cells around it. And chestnut blight is harmful. It starts secreting whatever effectors it uses to kill the surrounding cells. This is why in the trees, just the wild blight, we tend to see it at the branches where the branches join the trunk, because those are being blown by the wind and there's always little micro cracks forming, and also at the base of the tree where it's thickest. And you start to see blight set in when the trees reach the age when the bark ceases to be smooth like a beech and starts to be rough like an oak. That's when the cankers start to get in. In terms of the danger of moving blight around, there's plenty of blight on the wind in areas where chestnut was not present before the blight. There may not be so much around. You can bring it in on your tools. You can bring it in on your shoes. It's not a particularly airborne. There is an airborne form of the spores, but that's less common than the usual orange gloopy stuff that we see. So it could be done, but it's not like measles or COVID-19. You can get near a tree without giving it chestnut blight. You just should clean your tools and your shoes.
And just to say, put it a little bit more explicitly, so chestnuts, American chestnuts in particular, can get blight at any age. They can get it on the seed, they can get it in the seedling stage. It's just more likely, the longer that you're alive, the more likely you are to develop cracks or openings in the bark. And to Bruce's point, you know, these crotches and where the stem meets the a branch, or a bird lands on a twig and breaks the twig, and you know, spores get into a wound that way. That happens more often as the tree gets older. So we tend to say that chestnuts don't usually get the blight until they're 8 to 15 years old, and that's just because of various amounts of factors in terms of how openings happen on the tree, and as the bark starts to open up and mature. But seeds can get it. It's not likely, but it can happen. Well, those are all the questions I have. Bruce, do you have anything else you want to add to wrap things up? Okay. Well, thank you. That was excellent. I really enjoyed it. And I hope it was really informative to folks to know a little bit more about the molecular underpinnings of plant disease interactions. Next week, well, not next week, 2 weeks from now, August 7th, is that right? Let me make sure I get that date right. August 7th, we've got Stacy Clark with the U.S. Forest Service. She's been planting American chestnuts in the wild since, 2008, maybe even before. She's got some of the oldest outplantings of hybrid American chestnuts on U.S. Forest Service land. She's going to talk to us about restoration and some various aspects of getting these things in the ground, getting them to grow, and reintroducing them into the forest. So I hope you can join us on August 7th. 2 weeks after that, we've got Jean Romero-Severson from Notre Dame. She's going to talk to us a similar type of a talk. She's going to talk to us about metabolomics. So metabolomics 101, what is it? How do you know what it is? So we've got 2 really exciting things coming up, and of course more in the future. Every 2 weeks, join us for Chestnut Chat. With that, thanks for joining us on a Friday. Bruce, thank you again. That was excellent, and we'll see you in 2 weeks.
Bye.
Bye, everybody.